Welcome back, ladies and gentlemen. We will be continuing our discussion of gene expression today, specifically the beginning stages of transcription. Before we dive into transcription, let's take another look at the big picture. As mentioned before, the end result of gene expression is the creation of functional proteins. And in order to do that, we must go through transcription and translation. Last time we discussed chromatin remodeling, and this step takes place before transcription, and its purpose is to expose the DNA for the transcription machinery. So what is transcription? In short, it is the process of creating RNA using DNA as the template. This RNA, this RNA later on is going to be used for translation in order to create the proteins. But we're getting ahead of ourselves here. Okay. So first of all, though, what is RNA? Because we haven't talked about it before. So let's take a closer look. DNA is to the left and RNA is to the right. You see, for the most part, RNA is nearly identical to DNA. RNA is made out of 4 nucleotides, and they are connected by a phosphate backbone, just like DNA. However, there are several key differences. One of them is that RNA is normally single-stranded in our cells. Also, thymine, right? Thymine, which is a nucleotide in DNA, is replaced by a different nucleotide in RNA called uracil. The other 3 nucleotides are identical to the ones found in DNA. And this brings us to the final difference between DNA and RNA. DNA is deoxyribonucleic acid. Let me just write that down. So DNA, we went over this before. So DNA is deoxyribonucleic acid. And RNA is ribonucleic acid. And this difference in their names actually points out the difference in their chemical makeup. Here we have a typical adenine nucleotide found in DNA. You see, at this second position of the ribose sugar, right here at this position, the second position, we simply have an hydrogen in this case. If this adenine was part of RNA, we will have a OH group, a hydroxyl group instead. Indeed, the deoxy prefix in DNA, deoxy, no oxygen, is highlighting the missing oxygen, right? Makes sense. So, in summary, the 3 main differences between DNA and RNA is DNA is normally double-stranded, while RNA is single-stranded. In DNA, we have thymine as one of our nucleotides, but in RNA, it is replaced by uracil. And lastly, DNA only has a hydrogen at the second position in the ribose sugar of nucleotides, while RNA has an OH group, or a hydroxyl group. Now that we know what RNA is, let's talk about how it's made during transcription. Okay, so let's now begin talking about the transcription process. Be warned that there will be a lot of new vocabulary and terms I'm going to be throwing out. So let's approach this nice and slow. Remember, the whole point of transcription, is to make RNA copies of genes in order to serve as templates for creating proteins later on. The first step of transcription is called initiation. Let's say this dark blue region right here is a gene. Okay? And so after the chromatin remodeling, we have exposed this entire special region of DNA. Now, it's special because not only is the gene exposed, okay, not only because the gene is exposed, but the promoter region as well. What is a promoter? The promoter is a sequence of DNA, okay, highlighted in this this light green area. It's a sequence of DNA that marks where the transcription machinery will bind to in order to start transcription. I will talk about the different components of the machinery in a little bit. Um, the Tata box, which is also part of the promoter region, is a highly conserved sequence, which means it's found in many promoter sequences in the human genome, and not only in humans, but also other eukaryotic organisms as well. It's very well studied, but not every single promoter will have a Tata box. It's called a Tata box because the sequence for it is TATAAA. That's kind of the core sequence. There might be, there are variations of that with more adenines and such. The start point, which is also in the promoter sequence, indicates the exact DNA nucleotide that's going to base pair with the first RNA nucleotide. Okay, let me say that again. The start point indicates the exact DNA nucleotide that's going to base pair with the first RNA nucleotide in order to grow the RNA polymer. Again, I will show you what I mean soon. So right now we have a gene sequence over here to the right, and then we have kind of this promoter region to the left of it, right before it. The first components of the transcription machinery are these proteins. So just indicated in these blue spheres. These proteins are called transcription factors. Transcription factors. And sometimes you'll see these abbreviated as TFs. These are a large variety of proteins that come together and bind to the promoter region, forming these large complexes. The binding of the transcription factors recruits the other major component of the transcription machinery, called the RNA polymerase II. And often you'll see this abbreviated as RNA pol2. The RNA polymerase II is responsible for creating the RNA. And yes, there is a RNA polymerase 1 and 3, but they are not involved in gene transcription. Anyway, so this large complex of proteins, which I mean is the RNA polymerase II plus the transcription factors, together they are called the transcription pre-initiation complex. Let me just write that down. Transcription pre-initiation complex. And you'll see this abbreviated as PIC. Now the PIC opens up the DNA around the transcription start site. As you can see right here, we see this these two DNA strands kind of broken apart, right? So indeed, the nucleotides, the hydrogen bonds holding the nucleotides together, are broken apart to allow this shift from double-stranded DNA to two single-stranded DNA. This this region is called a transcription bubble. Let me just write that down. Transcription bubble. Transcription bubble. And so this is basically allows the RNA polymerase II to start synthesizing RNA. The first RNA nucleotide base pairs with the DNA nucleotide at the transcription start point, which is, again, I mentioned this before in the previous slide. So right here, we have the first RNA nucleotide base pairing with the DNA nucleotide at this region, the transcription start point. And the RNA grows, okay, to this direction, to this 5' to 3' direction. The RNA grows as more nucleotide base pairs with DNA and are added to the RNA transcript. I want to point out a couple of other things. Take note that RNA synthesis uses one of the two DNA strands. In this case, RNA synthesis is using this strand here, the bottom strand. Now, I'm going to use the word bottom strand and top strand just to describe to you, because this is how the image is laid out. Now, in reality, remember that there's really no top or bottom strand in DNA. These are just very arbitrary terms. So anyway, so this bottom strand is called the template strand. Template strand. And this top strand here, which RNA synthesis doesn't use, is called the coding strand. Now, why is this called the template strand? Well, it makes sense, right? Because in order to start transcription, in order to start making this RNA transcript, we must base pair the RNA nucleotides with the DNA nucleotides. And thus the RNA is using this bottom strand as a template, hence it's called the template strand. So now, why is this top strand of DNA called the coding strand? Well, first of all, it has the exact same sequence as the RNA that's being made. And the reason for that is because both this top strand of DNA and this RNA transcript that's being produced are complementary to the bottom strand of DNA, the template strand. Another way of thinking about this is that think about, what is this RNA transcript going to be doing? What is this RNA for? It's going to serve as code. Okay, it's going to serve as code to make proteins, and thus this top strand of DNA, because it has the same sequence as the RNA, can be called the coding strand. Of course, remember, the only difference is that in DNA we have thymine, and in RNA it's replaced by uracil. And I believe I already mentioned this, but I'm going to mention this again, is that remember that this RNA transcript is going to grow from the 5' to 3' direction, because it has to be complementary to the template strand. And here, the template strand, we usually write it from the 3' to 5' direction. Another name for the coding strand, or basically the strand of DNA that has the identical sequence, compared to the RNA transcript. Another term is often used, is called positive sense DNA. Okay, we can say the DNA has positive sense. And then so the template strand, the strand that the RNA uses to basically, for the synthesis, is sometimes called the negative sense DNA, or sometimes called anti-sense DNA, because it's complementary to the positive sense DNA. And in convention, normally when people describe DNA or they illustrate it in textbooks, they will write the positive sense DNA first on top. I guess if they were to draw out DNA horizontally, they will put the coding strand or the positive sense DNA on top, going from 5' to 3'. And then we have the antisense or the template strand or the negative sense DNA on the bottom. Of course, it's complementary, so it goes from 3' to 5'. Of course, you don't have to write it this way. It could be completely reversed. You can flip the DNA around and stuff. You can have the template strand on top. You can have the coding strand on the bottom. It doesn't really matter. It all means the same thing because you're not changing the DNA sequence or anything. You're just kind of changing the direction of how you look at it. But normally, the most conventional way of describing DNA, of drawing it out, it's like this. Again, with the coding strand on top going from 5' to 3', and the template strand at the bottom going from 3' to 5'. Phew, I know that was a lot to take in. So let's try to summarize what we discussed today. So we learned that transcription takes place after chromatin remodeling. And the whole purpose of transcription is to use DNA to make RNA, and this RNA will serve as a template for protein production, aka gene expression. Speaking about RNA, we learned that there are three main differences compared to DNA. First, DNA has thymine as a nucleotide, where RNA replaces it with uracil. In the ribose sugar at the second position, DNA has a hydrogen, where RNA has a hydroxyl or OH group. And lastly, DNA normally is double-stranded, while RNA is normally single-stranded. We also talked about transcription initiation today, the very beginning of transcription. We learned that chromatin remodeling exposes a very special region of DNA, namely the promoter region, and right after that will be the gene sequence, exposes these two areas of DNA. Exposure of these areas allow transcription factors to bind to the promoter region, and then that leads to the binding of RNA polymerase 2. And transcription factors plus the RNA polymerase 2, they form a complex known as the transcription pre-initiation complex, or the PIC. The PIC then converts the promoter region from a double-stranded area to a single-stranded area, and kind of breaks the hydrogen bonds that are keeping the base pairs together, forming this transcription bubble. And after that is when RNA synthesis can first begin at the start point. The start point just refers to the area that refers to the DNA nucleotide, in which the first RNA nucleotide base pairs with, in order to start growing the RNA transcript. This takes place on the DNA, on one of the DNA strand, specifically is called the template strand. And the other DNA strand that RNA synthesis doesn't use, it's called the coding strand. Again, it's because it's complementary to the template strand, and thus it has the same sequence, not counting, the difference between thymine and uracil, of course, has the same sequence, identical sequence as the RNA transcript being made. Okay, well, that pretty much wraps up this lesson for today. I hope you enjoyed it and you learned something new. And then next time we're going to talk about the rest of transcription, the elongation and termination steps. Okay, well, thank you everyone for stopping by, and please like my videos and subscribe to my channel. It really means a lot to me. Well, until next time everyone, I hope you all have a great day.